Welcome back to O-Level Oasis, your go-to place for preparing for the O-Level Biology 5090 exam. We are currently studying Chapter 18, which focuses on biotechnology and genetic modification. Today, we'll begin Unit 18.2 on genetic modification and go through each specification step by step together. You'll find the topical notes linked in the description below, so make sure to check them out. Now, let's get started. We begin with Specification 1, which asks us to describe genetic modification as changing the genetic material of an organism by removing, changing or inserting individual genes. So, we define it as, genetic modification is the process of changing the DNA of an organism by adding, removing, or altering specific genes. In simple terms, it means directly editing an organism's genetic material to give it new or improved characteristics. Genetic modification is a scientific process where the DNA of an organism is deliberately altered. This can involve adding a new gene from another organism, removing an unwanted gene, or modifying an existing gene to change how it works. These changes are made to produce useful traits, such as better growth, resistance to diseases, or improved nutritional value. The process is carried out using genetic engineering techniques, where scientists carefully identify and transfer specific genes. Once transferred, the new gene becomes part of the organism's DNA and can be passed on to future generations. An organism that has been altered in this way is called a genetically modified or GM organism, and if it contains genes from a different species, it is known as a transgenic organism. When DNA from two different organisms is combined, it is called recombinant DNA. This allows the organism to show new characteristics that it would not naturally have. Genetic modification is widely used in areas such as agriculture, medicine, and scientific research to solve real-world problems like increasing food production, developing vaccines, and treating genetic disorders. Now, looking at how this is done using bacteria, the process begins with selecting the required gene, such as the gene responsible for producing human insulin. This gene is then isolated by cutting it out using restriction enzymes, which create sticky ends. A plasmid, which is a small circular piece of DNA found in bacteria, is also cut using the same restriction enzyme so that the ends match. The desired gene is then inserted into the plasmid and joined using DNA ligase, forming recombinant DNA. This recombinant plasmid is then transferred into a bacterial cell. As the bacteria reproduce, they copy the plasmid and begin expressing the inserted gene. Finally, the bacteria are grown in large numbers in fermenters to produce large quantities of the required protein. As you can see in the diagram, the plasmid and the gene of interest are both cut using the same restriction enzyme to create matching sticky ends. DNA ligase then acts like a molecular glue, joining the gene into the plasmid to form a recombinant plasmid. This recombinant plasmid is inserted into a bacterial cell, where it can be replicated or used to produce the desired protein.